All right, good afternoon, everybody, and thank you for coming to our symposium on Microbiology, the Built Environment, Implications for Health and Design. And thank you to the Alfred P. Sloan Foundation for funding our symposium today. So before we get started, just a couple things. If you would be able to please fill out the sign-in sheet. There's one right there, and I think one up here, if you could pass those around. Just so we know who's here. And also there's a survey on your chair, so if you could please fill those out for us at the end so that we know what you think of the symposium. Also, you've probably noticed that the symposium is being filmed for future dissemination, so those people who couldn't be here today will be able to view it in the future. Finally, we do have an announcement about an exciting future event that's going to be held here in DC in October, and I'll let the chair of that event, Dr. Lynn Schrimmel, tell you a little bit more.
Everybody. We thought this would be a great time to bring up our Sloan-funded Microbiology of the Built Environment. This is end of a 5-year and sometimes 10-year program. We'll be at the National Academy of Sciences next October. It's a 2.5-day event. It's open registration. It'll be open in March. If you go to microBEnet, this is our home site for the project. There's information about the meeting. I have handouts I'll put at the back. We encourage you to attend. It's going to be about 300 participants. From across engineering, architecture, microbiology, infrastructure. So we're very interested in having a broad participation. We thought this would be a great opportunity to talk to people about it. So if you have any questions, we can chat afterwards.
All right, thank you very much. And so for the format today, I'm going to give just a quick, maybe 4 or 5 minute introduction, and then each of the 5 speakers is going to give about a 12-minute talk, and then we'll have a panel at the end. So we'd love to hear your thoughts on what we're talking about, as well as any questions. And we ask that you save any of your questions until the end. So we spend 90% of our time indoors. Think about where we are today. We're spending most of our day in a hotel in the built environment. And this is actually a pretty diverse and interesting ecosystem that we're really just starting to learn about. So you can look at this example graph over here, which shows 40 homes in North Carolina that we're collecting. You can see unique but diverse microbial communities on even just the different surfaces within this home. So you can compare the toilet seat to a pillowcase to a refrigerator and the door trim and see how these microbial communities might differ. So we're going to talk a lot about these kinds of things today. This is a really exciting time to be doing research in microbiology in general. The conference with the same theme for NCSE was held 10 years ago, and at that point, the cost to sequence one human genome was here about $10 million. Today, the cost to sequence one human genome is around $1,000. And so we can use these sequencing technologies to study microbial communities as well. And the benefits of using sequencing technologies to study microbial communities is that you can get a comprehensive picture of all the fungal and bacterial species that are present in a system. Previously, we were using more traditional culture-based methods, which only get a small snapshot, a small subset of the microbes that are actually present. So now we can get this more comprehensive picture. We can do it much more inexpensively and get this really exciting data. So around 2007, around the same time, we also, NIH launched the Human Microbiome Project, and the Alfred P. Sloan Foundation in 2004 launched their Microbiology, the Built Environment program. And they've since invested about $50 million into this program to learn more about the microbial communities that surround us every day. And so a lot of exciting work has come out of that. You may be familiar with some of the human microbiome work, and that the human microbiome has been linked to a very wide range of diseases. So just look at this list of diseases for a minute. We have things ranging from allergies and asthma to obesity, colon cancer, psoriasis, dental caries, and many others. So the human microbiome, the microbiome in ourselves, has been linked to all these diseases. And what we're going to talk about today is how environmental exposure could potentially impact that microbiome and also impact that disease directly. So we're going to focus on the indoor microbiome and how that might influence human exposure and can impact disease, and also about the design aspect. So when we think about designing buildings and we think about the choices that we make every day, how does that influence our indoor microbiome and eventually potentially disease? So we have some really great speakers for you here today. So I'm going to start out talking about my work with childhood asthma and indoor fungal exposures. Next, we're going to have Dr. Michael Morowitz, who's going to talk about the impact of a hospital microbiome on neonatal development. Next, we'll have Dr. Amy Pruden, who's going to talk about her work on premise plumbing systems and the Flint, Michigan water crisis. After that, we'll have Dr. Jade Mitchell, who's going to talk about her work on antibiotic resistance genes in the indoor environment. And finally, Dr. Mark Weir, who's going to talk about quantitative microbial risk assessment and assessing the risk of the microbes in the indoor environment. So first, with my talk today, I'm going to talk about the indoor microbiome and linking these indoor microbial exposures to health outcomes, in particular childhood asthma. And I focus particularly on fungi, so that's what my talk is going to reflect today. Part of the reason that I focus so much on fungi is that the exposure of asthmatics to mold in housing costs an estimated $3.5 billion per year. And it's not really as pretty as this nice piece of artwork would have you expect. So we know that dampness and mold in housing is associated with these negative health effects, especially in these populations of asthmatic children, which make up 8% of the population. But we're still working on identifying the exact causal agents and how this mold in housing does actually cause this asthma. So one place that we're going to look is actually in the house dust itself. And the reason for this is that because that's where a lot of our exposure comes from in the home environment. So you can look at these graphs, which show 6 cities around the world, and they show the indoor-outdoor ratio of bacteria and fungi under occupied in black and unoccupied conditions in white. And you can see that under the occupied conditions, the concentrations of fungi and bacteria increase substantially. And most of this is actually from resuspension from the floor dust. So 83% of airborne bacteria in the air comes from floor resuspension under occupied conditions, and 66% of airborne fungi comes from this resuspension from the floor. So that's why in my talk I'm going to focus so much on that floor dust as an important source of human exposure. So I'm going to highlight 2 studies today. The first is an epidemiological study, and the second is a laboratory-based chamber study. So the first study that was done was to look at asthma severity. So in a vulnerable population of asthmatic children, try to characterize their indoor microbial exposures and how that influenced their asthma severity. This was part of the STAR study done in Connecticut and Massachusetts with my colleagues here, and was a prospective study done where we collected dust at the beginning of a month and then recorded asthma severity for that child over the remainder of the month. And we included 196 children. Asthma is a fairly complex disease and has different subtypes. 2 subtypes that we considered here are allergic and non-allergic asthma. Allergic asthma is mediated by eosinophils and is an IgE, involves IgE as an allergic type response, the more traditional type of asthma that you might think of. More recently noted is non-allergic asthma, which is mediated by neutrophils and is an acute inflammatory or more of an irritant type response. So we divided the children into this study into those with allergic and those with non-allergic asthma. And what we found is that the indoor microbial communities influence these children's asthma severity differently based on their asthma subtype. So if you look at the top row of this graph, you can see that for the allergic children, we looked at the fungal communities they were exposed to, basically what they were exposed to. Are they exposed to allergenic fungal species? And we saw that this influenced their asthma severity. We looked at fungal concentration and didn't find an association. So it didn't necessarily matter how much they were exposed to, it was just what. For the non-allergic kids, we looked at the fungal communities and it didn't matter what they were exposed to, but if these kids with the more irritant type response were exposed to high concentrations of fungal communities, then they had increased asthma severity. So just to recap, for the allergic children, it mattered what they were exposed to in their indoor environment. For the non-allergic children, it mattered how much they were exposed to. The nice thing about using methods such as next-generation DNA sequencing is that you can get lists of fungal species that these children were exposed to, and I'm not going to go through this whole table, but there's a couple interesting things that we found. On the left is, and this is for all the allergic kids, on the left are species that were potentially detrimental for asthma severity, on the right, those that were potentially protective. Only one of them was actually statistically significant, and that was Volutella, which is sort of interesting, sort of an unexpected result. But we have a couple other things that were not quite statistically significant that showed up as potentially interesting. And this was potential associations with yeast, so Cryptococcus, which with different species showing up on the potentially detrimental and the potentially protective side, and Condoa, another type of yeast, and both of these had showed up previously in another one of our asthma development studies as potentially protective. So we can't make any strong conclusions here, but it's something for potential future research. The interesting thing about Cryptococcus is that it's fairly difficult to culture, so it's only recently been examined more in depth with the use of these new technologies. And it's been shown in another epidemiological study as well as a rat model to potentially be associated with asthma. So it's something to keep in mind. And this is just what the Cryptococcus in Condoa. Condoa is a fairly rare fungal species. In this study, it was only present in 13 homes, but something to keep in mind for the future. So for this epidemiological study, we saw for asthma severity that the associations between fungal exposure in the indoor environment were dependent upon asthma subtype. So for the allergic kids, it mattered what they were exposed to. For the non-allergic kids, it mattered how much. And we see this interesting association with yeast showing up that deserves some further study. The second part of my talk that I'm going to move on to is a laboratory-based chamber study. So you can see these lovely pictures up here of mold growth growing on damp building materials. We're all familiar. We've probably all smelled, for instance, a damp building where you walk in and smell that musty odor, and you know that microbes are growing on these building materials. But we wanted to ask the question about the contribution of growth under more normal conditions. And our question was, because resuspended dust is such an important source of exposure, we want to know if growth can occur there. So our question was, is relative humidity in the air sufficient to support microbial growth in carpet? So we decided to collect carpet and dust from a home. So this was dust that was actually already sort of seeded. It was a 10-year-old worn nylon carpet, and expose it to different relative humidity conditions in laboratory chambers and see what grew. So our methods look something like this. We take a 10 by 10 carpet coupon, apply dust from the same house, embed it using a modified ASTM method. I must admit I'm impressed. ASTM has a method for just about everything, including embedding dust in carpet. Once the dust is embedded, we place the carpet in our relative humidity controlled chamber. And these did equilibrate fairly quickly if you measure the water activity of the dust, which is related to the relative humidity in the chamber. And what we found is that, yes, the microbial communities grew in these carpets based on the relative humidity in the room. So these top two graphs are 1 week long experiments. So you have fungi on the left, bacteria on the right, the relative humidity in the chamber on the x-axis, and spore equivalents per milligram of dust on the y-axis. And we see our fungal growth starting right around 80% relative humidity compared to the OD, the original dust. And about a 27 times increase in fungal concentration at 100% relative humidity. We also see bacterial growth, but only occurring at 100% relative humidity and not at the lower levels. We did some longer-term experiments out to 6 weeks at 100% and 85% relative humidity. At 100%, we actually had to end the experiment after 3 weeks because the carpets were so covered in visible fungal growth, we didn't want to continue. But at up to 6 weeks at 85%, we still see continued fungal growth and even the potential for some additional growth in the bacteria. So these are fairly high relative humidity levels for a building. You wouldn't generally expect to see these on a normal basis unless you have a dampness problem or you're maybe in a hot, humid climate in the summer. But this is an interesting first result to continue on with. We see changes in the microbial communities, and in fact, the moisture is the largest driving factor. These are principal coordinate plots where similar microbial communities cluster together, and you see a clear trend in the fungal plot from the original dust to 100% relative humidity. And for the bacterial communities, everything clusters together and starts to separate out at 95 and 100% relative humidity. So what does this mean for exposure? Well, actually, if you look at the fungal exposure, we have our background exposure that you'd expect to see, and this is a fairly rough model in orange for the fungi and the bacteria, and then the additional exposure you might expect to see from this growth. And in fact, I don't recommend anybody leave their house at 100% relative humidity for a week, but this is a potentially important source of human exposure that we need to consider is the actual microbial growth in the dust. We're currently working on experiments to make this more realistic in terms of building conditions. So please come check out our posters. We have a poster about detection and also a poster continuing on this work, 7 and 47. So go check those out if you get a chance. So for this second study, we saw that relative humidity did influence the microbial growth in carpet with a wider range of growth for the fungi than the bacteria, which is fairly expected, and that this is a potentially important source for human exposure that we need to consider in the future when we need further work on this. For future directions, it's important to consider the indoor microbiome and its impact on human exposure for both the asthma severity study that we talked about and this fungal growth study to consider this human exposure. This also has potential policy implications if you're thinking about mold in housing and potentially using these values to actually calculate out a model for what this would mean for human exposures and dampness in the environment. And finally, one of the big questions out there is, what does a healthy microbiome look like? And we're making a lot of strides forward to help figure this out with all the research that's going on, but it's still a question that we need to continue to address. So I'd like to thank the Alfred P. Sloan Foundation for funding this work, as well as NIEHS, all the colleagues and co-authors on the project, and the study participants. So thank you, and I'll give it over to Dr. Michael Morowitz. Oh yeah, and we'll take questions at the end.
Thanks, Karen. Okay, my name is Mike Morowitz. I'm from the University of Pittsburgh. I'm a general and thoracic pediatric surgeon. And I have a research interest in the intestinal microbiome, which is a term for intestinal bacteria, but as well fungal organisms that I'm not as familiar with, and viruses and other types of organisms as well. I'm particularly interested in the intestinal microbiome for critically ill patients. I'm interested, our lab is interested in adults, children, and then the focus of this particular talk, premature hospitalized infants. And one of the reasons that my research group and others have been interested, and probably you all also are interested in this topic, has to do with the very simple and somewhat disturbing fact, which is that if I were admitted to the hospital today, and I was admitted to a hospital room where the prior person that had been in that room had had an infection, I would be at a clear increase in risk of developing that very same infection relative to somebody admitted to another hospital room. Very disturbing concept, which was addressed in a recent review article, and I included a quote here, which I think was thought-provoking from the authors of this article. They said, for a layperson, it would be difficult to explain why, just because the last person in the room had an infection, the new admission has an increased risk of acquiring that same organism, particularly since additional cleaning measures are often in place when the infection status of a patient is known. And that's absolutely true. We take extra precautions when somebody's infected, so you would think maybe that would make it even less likely to propagate an infection. What I didn't know very much about, and those of you in the audience I'm sure know way more than I do, is how difficult it is to clean hospitals. And one particularly interesting study was published a few years ago from Australia, where a particular hospital was decommissioned. And some enterprising investigators from this study, before and after the hospital was decommissioned with various cleaning processes, looked at hospital surfaces, and what they found without a doubt was that there was widespread colonization of hospital surfaces with the same organisms that are really problematic in causing hospital or health care acquired infections in patients around the world. So our group has been fortunate to be receiving support from the Alfred Sloan Foundation through the MicroBEnet initiative. To my knowledge, there are 3 projects associated with this initiative, specifically studying the hospital microbiome as opposed to other indoor environments. Our interest is in how the indoor hospital microbiome may or may not be linked to the human microbiome, and as I said, in particular, premature infants. We became interested in this topic a few years ago when we began using some of the molecular DNA sequencing techniques that Karen referred to study fecal samples from premature infants. And we were surprised, other people have done this work too, we were surprised, and I think others have been as well, to see that the organisms that are present in fecal samples from a newborn premature infant, I'm talking about in the first days and weeks of life, are really dominated by pathogenic species of bacteria. And that's even in healthy babies that are doing quite well. This particular graph, on the x-axis are the days of life that one particular baby was born, and as is often shown in these types of studies, the different colors represent different types of organisms, and many kind of bad actors are present in this particular infant's samples, and this infant actually did great and had no problem. It's known, and in some cases suspected but not yet proven, that there are some particular factors that impact which organisms are present on which babies and in samples from which babies. Same with us too. But as it pertains to babies, mode of delivery, vaginal delivery versus cesarean section is important. Type of feeding, breastfeeding versus infant formula. Antibiotic exposure, very important as you know. And we were interested in the link to hospitalization, the actual physical environment in which a hospitalized baby is staying. The clinical relevance of this problem is significant because it has been shown for at least 2 important comorbidities of premature infants that I've listed here, necrotizing enterocolitis and sepsis, but probably others as well, that outcomes can be associated and possibly caused by abnormal or aberrant types of bacterial colonization. And so that's our interest in this work. And I want to say at this point that everything that I've done on this project and other projects has been in very close collaboration with a very brilliant woman named Jill Banfield, who works at the University of California, Berkeley. Shown here, back here, is a PhD student who now has his PhD linked to this project. His name is Brandon or Bubba Brooks, and here he's pictured having spent time in Pittsburgh from Berkeley, California, working on this project. I won't go over this. This is a slide representing what Karen also referred to about sequencing-based techniques to do this work rather than culture-based techniques. In particular, the work that we've done is of the sort of category called metagenomics, which allows for very deep study or analysis of the bacteria that's present. The next couple slides, the details are not very important, but the take-home message are the following. When you do very deep molecular analyses of bacteria, you can learn not just the names of the organisms, but important genomic characteristics of these organisms. For example, are they virulent, predicted to be virulent? Do they have antibiotic resistance determinants? I think we'll hear more about that later. But the point of this slide is that when we saw different organisms, over here are 10 different infants from one particular study, different rows on this heat map represent different strains. And the important thing is, until recently it was thought that if you and I have E. coli in our samples, those E. coli are probably very similar. But in fact, the E. coli strains can be quite different. And this fact is particularly true to pathogenic types of species that can change their genomes in certain settings. And an amazing thing that we found in one particular study of 10 infants was that we looked at all the strains of bacteria that were found. In this study, we found 149 strains of bacteria in 10 infants. And of those 149 strains, only 4 were seen to be shared across infants. That means we have very personalized microbiomes, just like we have personalized genomes. But then when we looked at another study from another time period, one interesting thing came out to us, which is that over time, still not many strains are shared across individuals, but a few small, quote, persister strains could be seen in the neonatal ICU, shown by these black boxes on the heat maps. And so that indicated that some strains were persisting in our ICU for several years. And then the question addressed by this work with the Sloan Foundation is, where are these persister strains coming from? Are they related to the indoor environment? The first pilot study that we did of 2 infants, involved looking at a number of surfaces in the neonatal ICU, and we saw clear evidence that, in particular, sinks in the ICU were sharing the same organisms with the fecal samples from infants, and in many cases we could see that they would appear in the sinks specifically prior to infant fecal samples, which are shown up here. This subsequent work through the Sloan Foundation has been wider, deeper, larger sample size of infants in ICU rooms. Shown here is simply the amount of bacteria, not the types, but higher points on this graph indicate larger amounts of bacteria. The takeaway point here was that the sink, the second item here, has a lot of bacteria, particularly around the drain in the sink. Other surfaces, such as those that are touched, we looked at nurses' cell phones, the beds, other furniture in the room. They have important amounts of bacteria, but lower. A little fun fact that came out of this study was that the day of the week was relevant. Actually, Mondays had the highest amount of burden of bacteria around the sink drains than any other day of the week, perhaps related to cleaning practices or visitation in the room. When we looked at the types of organisms that are present in the indoor microbiome, the surfaces that are touched by nurses, visitors, are very clearly human skin bacterial divisions, specifically and most commonly Propionibacteria and Corynebacteria. We can talk more about that if you're interested. The sinks were very different, though. They tend to have gram-negative pathogens, which are common in fecal samples from babies, but also adults that are in the hospital. These are not organisms that you typically see in healthy individuals. And then to sort of close the loop on what I've been talking about, I can distill this down to say that the x-axis of these graphs is basically a year of sampling, and this is just meant to say that a particular strain of bacteria could be seen at the beginning and the end of this year when we looked at various samples in the ICU room. Then the key question is, well, do the persister strains in the infants match up with the persister strains in the ICU environment? And that is what we see. Shown here is a list of several strains of bacteria, only those that have samples seen across infants and across time. And when you display these next to down here are organisms that persisted in room samples, particularly the sink. What we found are that organisms that are seen over time in multiple infants are the same organisms that can be seen over time in the ICU room environment. A few things that I didn't have time to discuss, but is the fact that actually many strains in infant samples could be seen in the room, but they're usually specific to one infant or one ICU. And directionality actually goes both ways. Sometimes we see it in the baby first, other times in the room first, but there's clear crosstalk. And so I'll summarize by saying that the gut microbiome of premature infants is enriched with pathogens. The NICU room microbiome is enriched for skin organisms on touch surfaces, but also gram-negative pathogens in the sink. The crosstalk there appears to be bidirectional, but we believe that the NICU room environment, at least evidence suggests that the NICU room environment is a reservoir for these persisters. Future directions would include real-time monitoring both of the patient and the room while the patient is there, and microbiome-based therapies or interventions, possibly for the baby, but also things that you all are experts at, next-generation strategies for cleaning the rooms. Like I said, I don't know much about it, but I'm eager to learn. And I would like to acknowledge members of my lab, collaborators elsewhere, but as I mentioned, particularly the lab of Jill Banfield and Brandon Bubba Brooks. We also worked closely with Dr. Bill Nazaroff at UC Berkeley, who is an expert on indoor air microbiome, and he was a key participant to these studies as well. So thank you for the invitation to participate and for the chance to present our work.
Well, thank you so much. It's a pleasure to be here, and thank you for the organizers for this event. So we've heard a bit about mold and hospitals, so now I'll scare you a bit talking about your water. I first want to acknowledge my co-PI, Mark Edwards, and also Anisha Patel and Joe Falkenham and Annie Pierce that have been working on this project, and of course the Sloan Foundation for funding this work. So I'll talk a little bit about this proof of concept of what we've been working on, looking at the microbes in your tap water, and it turns out there are a lot of them, and we've been looking at the potential to intentionally control the kinds of microbes at your tap. And the key to this is largely the water chemistry as well as other physical chemical factors. And I'll make the case that if we don't start admitting to ourselves the reality that the water that we drink and bathe in daily is full of microbes, then we're setting ourselves up because we're going to be unintentionally shaping the microbes that are there. And I'll make the case that we should try to be mindful and intentional about this, or we will have unintended consequences. And I want to touch a little bit on how this might affect how we conceptualize risk. So, in case you're not aware, our water infrastructure is in shambles. It has been for well over a decade or longer. The ASCE has been telling us that with this report card where it gets around a grade of a D. But I think an example that really put this in our faces is what happened in Flint, Michigan. People were poisoned by their tap water. And I'm glad that this was brought to the forefront. And scientifically speaking, it was actually quite simple. It was water chemistry. It was a switch in water chemistry caused the lead from these old lead pipes to leach into the water. So this gets us thinking about the importance of water chemistry and being aware of water switches. And here's essentially what we saw is that the Flint River water was high in chloride. It's salty, it's more corrosive. Very simple experiment showed if you put an iron nail that the water turns rusty, whereas if you do the same with the regular Detroit tap water, the water is fine. And these citizens here figured that out right away, and unfortunately it took a very long time to get the attention of people to do something about that. Now, besides the lead, you might not have heard as much about something else that was going on. Legionnaire's disease. Legionnaire's disease is caused by infection of the lungs, usually by inhaling water aerosols. So it seems no small coincidence that they switched the water and you went from this baseline level of Legionnaire's disease in the population and suddenly it spiked up. And so 92 people became ill and 12 people actually died during the period of the Flint River water usage. And Mark Edwards and I had been working for some time through our Sloan Foundation grant just on basic understanding what are the microbes in your water and what triggers Legionnaire's disease. And so it just was strange to watch this in the real world unfolding. Our hypothesis, essentially, that if you have higher iron in the water, that can trigger Legionnaires. So this was a first example of where we saw corrosion at a citywide scale linked to Legionnaire's disease outbreak. So iron is a nutrient for Legionella, but it also reacts with chlorine and causes that to decay very rapidly, and then you lose that protective barrier against microbes. Now, the good news is, here is the water a year ago in a hospital sink. Doesn't look too hot, 0 chlorine. And here's what it looked like when we went back to that same hospital sink a year later. So switching back to the Detroit water has really improved the water quality. So we have an experiment going on in the lab now to try to understand exactly what it was about the Flint River water that triggered these Legionella bacteria to grow. And so we're simulating your household water heater. These are glass bottles in a hot room. And Legionella and other pathogens that grow in water systems tend to like this warmer environment, like a hot water heater. And we did replicate the basic phenomenon that very low Legionella, it died off quickly when we put it in the Detroit water, but it's been thriving in the Flint water. But some good news is that our condition with copper pipe tends to have lower levels of Legionella. So it's giving you a sense of this interplay between the water provided by your utility and things that you can do at home as a homeowner to protect yourself. So copper is a natural antimicrobial and something we'll be looking into in more detail. Now, bigger picture, how did we get started on this journey? As I mentioned, we were just asking the fundamental question, what controls the microbes that come out of your tap? And so you have this interplay between the water delivered by the water utility, and it passes through miles of pipe through a service line to your property. Now, most regulations end right here at the property line. And it's kind of tough luck for the consumer to figure out all these complexities for themselves. And that's where exposure happens. Like I said, Legionnaires is something, it's a bacteria that's in aerosols. You're exposed to things like showering. So what do we do about this? Well, digging deep into the microbiology, this is a cross-section of a pipe. We can try to understand more about what triggers these bacteria to grow. So if you've had any plumbing work done, you'll see that it's not always pretty what your pipes really look like. You've got this slimy layer called biofilm, lots of microbiology going on, nutrient cycling. Drinking water's pretty clean. We remove pretty much all the organic carbon and nitrogen, but microbes are smart. They find ways to scavenge what's in there. And plus, if you're in a biofilm, you just sit and just the water flowing is bringing that food to you. The pipe material, as we saw in the case of the copper in the Flint experiment, that can be important. That could inhibit microbes, while plastic materials can leach carbon and feed microbes. And water heaters, the anodes, release hydrogen. That is an energy source for microbes. So all sorts of interesting complexities going on in the home that we're trying to understand. Now, backing up, what about the water delivered by the water utility? So we used this next generation DNA sequencing at 5 different utilities across the nation, and we saw 5 different clusters of microbes. Even though we were using a controlled rig to simulate the home so everything was the same, it turned out that it was the utility controlling the baseline types of microbes. So that's important because that gives municipal control. Inside the water heater, the temperature can be very important. If you turn it up, you can kill pathogens, but you can also increase your scalding risk. So we did an experiment here led by our student William Rhodes, where he constructed a pipe rig and tested different water heater settings and connected it to these pipe rigs, which is important because you have to realize that the water coming out of the tap is not the same temperature as the water heater. It cools down. So you don't necessarily have that protection of the thermal disinfection at the tap. And so what we saw is quite interesting. We saw that in 2 rigs that were replicated, that the warmer temperature had more Legionella, okay, but up to a point, right? Once you got it hot enough, you killed it off. When you had higher flow rate, you were actually delivering more nutrients, more carbon and nitrogen, so we started to get higher levels of Legionella that way. And very interesting was we found this sweet spot where if the water heater temperature, in this case 51 Celsius, was not hot enough to quite kill the Legionella but just shock them, they actually recovered quite well and really became dominant in the population because it killed everything else off. And then this shows what was going on with the broader microbiome. We saw in the bulk water there was a threshold at hot temperature. Suddenly the composition changed, whereas in the biofilm, as the temperature increased, it was more gradual. And then we're also doing some metagenomic work. So this is a little different. Instead of just looking at the 16S gene that identifies bacteria, we shotgun sequence all the fragments of DNA, and that's nice because you can see pathogen markers, virulence genes, antibiotic resistance genes is something we're interested in. And basically, this is another way of looking at the data, but we saw the same trends with shifts in time and temperature. So the metagenomic data validated the microbiome data. But it gives us a deeper look into functions, and we saw functions that we were interested in, like this hydrogen oxidation and the nutrient cycling. And then I just want to mention one other very important water chemistry shift that's going on, and that's recycled water. So this is one way of advancing water sustainability, right? We have drought, tight water resources. So you take sewage and you treat it to an extremely high standard, probably cleaner than anything you would typically encounter in a river or lake, but it's still a very different water chemistry and something that maybe our pipes have not seen. And so we're surveying paired potable water systems, traditional potable water, and recycled water systems to see how that's affecting the microbiome where at the potential point of exposure, because that's where the risk could be. And so irrigating golf courses and recreational fields. And we are looking at antibiotic resistance. So Legionella, other opportunistic pathogens, but antibiotic resistance as well. And all of these have in common that, you know, they're growing concerns, and they're things that can grow in the water system itself. And with the metagenomic methods, we can look directly at the antibiotic resistance genes and see if switching these water sources is going to have an influence. And so in the interest of time, you can ask more questions at the end, but we're looking at things like the water treatment. Are there different ways that we can treat the water and present barriers to the spread of antibiotic resistance genes? And then this is an example of our metagenomic data. So when you distribute recycled water and look at the point of use, you see them increasing because of growth in biofilms. But it's important to put it in context. You can see that there are also antibiotic resistance genes in our regular plain Jane potable water. So we need to get a handle on what are the normal baseline levels of antibiotic resistance genes, which are the ones to be concerned about. The biofilm comes into play again. We see that they tend to partition there. And then finally, another big change that we're looking at is with green buildings. So green buildings, you have water conservation, which is good, but conservation inherently means stagnation. So water sitting longer at warmer temperatures, more microbial growth. This needs to be looked at. And so in terms of risk, we need to think broader. One concept is the exposome concept, thinking about gene transfer, not just individual pathogens or surrogates, and thinking about how it influences the human microbiome. So we're delivering thousands of microbes at the tap, we're drinking that water, we're bathing in it. What kinds of implications does that have for microbiome diseases? And what about emerging concerns like antibiotic resistance and opportunistic pathogens? So, thank you very much.
Thank you to the organizers and to the Sloan Foundation for inviting me. I'm going to talk about antimicrobial resistance and risk assessment. That's hard to see. I'm not going to read the statistics. We know that antimicrobial resistance is a big problem in terms of health outcomes and economics. We also know that, thanks Mark, that antimicrobial use in animals is one of our largest consumer of antibiotics, and as a result may result in resistance in human disease. So a lot of the focus of the research has been here in terms of understanding resistance from agricultural use in animals and how that translates to human disease. There have been some previous funding commitments in this area that focus on antibiotic stewardship, strengthening risk assessment, surveillance, and reporting capabilities. But in my talk, I want to try to emphasize where there are still data gaps. And I should say that these efforts are across a One Health approach. So considering veterinary medicine, human medicine, and the ecosystem. In terms of environmental pathways, there is a lot of data that has looked at environmental compartments, those that are listed here, and identified several hotspots or reservoirs for antimicrobial resistance in the environment. But for risk assessment, we really need to tie these compartments together so that we can build exposure models for humans and estimate those exposures where they're exposed in order to do risk. This is our general paradigm for quantitative microbial risk assessment. We identify the hazards. We want to evaluate using monitoring and modeling data what the expected exposures are and estimate exposure doses. And then we want to combine that with a dose response model that predicts risk based on those exposure doses. And then in risk characterization, we tie this together with an understanding of the variability and the uncertainty. For antibiotic-resistant bacteria, we ultimately want to know what is the risk of treatment failure in human medicine. And that goes at least one step beyond just the response of infection, illness, or death. So for antimicrobial resistance, it complicates the risk assessment framework, and as Amy mentioned, we need a new framework. In terms of hazard identification, we have to look at the resistant bacteria, the presence of genes, as well as the chemicals that may have selection pressure. Excuse me, my throat's doing a weird thing. In terms of exposure assessment, there's this potential for horizontal gene transfer. So we don't just look at uptake of the single pathogen, which is generally how we do microbial risk assessment. But now we have to consider uptake, mutation, and in multiple environments. In humans, in the gut, as was discussed, in the environment, and in animals, since there's interaction between all three of these environments. In terms of dose response, we need to consider also modulated dose response. And by modulated, I mean that in the presence of selection pressure, we don't expect the response to be the same. For risk characterization, there are cumulative risks that need to be identified. So our typical risk assessments for food safety, which is one area where it's well established, would look at farm to fork. In this case, we would go farm to fork to failure. But when we start considering the indoor environment, or the environment in general, there's some interplay that change the exposure and the dose response, starting at the farm level, but all the way across the paradigm to treatment failures in hospital settings. So one study that we recently analyzed was provided by Dr. Bo Norby in veterinary medicine at Michigan State. He had collected 24 feedlot steer, for 24 feedlot steers, a set of fecal samples over a period of 26 days. And his purpose was really to evaluate the timing of treatment and the emergence of resistance in order to evaluate some dietary and environmental effects, but also other epidemiological factors like age. We found when we looked at these samples that, I should say first that the time period is really not well established for when we should look for the emergence, and even after the emergence, when we would return to some baseline. But we looked at 0 and 9 days, where these cattle were dosed at 0 and 3 days. And we did a comparison. But the more interesting thing here was that some of these cattle were kept in their home environment, which we're considering a transition pen, because this happened during the lactation cycle. And the other cattle were taken to what we were calling a hospital pen, which is generally considered clean. It's sanitized and there's new bedding. So when we look at, say, the green and the yellowish green in comparison, we're looking at hospital versus home, and then we also can compare at the 9-day time point the orange and the red, which is hospital versus home again at 9 days. Depending on the antibiotic that resistance was selected for, it really varies whether there is an increase or a decrease in the abundance of genes. Overall, and this was contrary to our hypothesis because, again, the hospital pen is clean, which you alluded to, we found that there was higher resistant coliform compared to the home pens. And then we also found that in the hospital pens there was higher abundance and diversity. I'll ignore the fact that we decreased over the 9-day because that was also contrary to the hypothesis, but we actually think that what we were measuring were bacteria that were resistant to the antibiotic ceftifor, and that's why we saw that decrease. So this whole concept of clean comes up. Recently we've done some work looking at surfaces that would be found in hospital settings for mica and steel, and their persistence and inactivation based on UV in order to try to understand persistence better. So we know that antimicrobial resistant bacteria are susceptible to sanitation and to disinfection and even TUV. But really our understanding of this at a pathogen-specific level is not very well understood. So the pathogens that we looked at were Clostridium difficile, which is associated with antibiotic usage, MRSA, which is resistant, and vancomycin-resistant enterococcin. We found that initially what you would expect to achieve, like around 3 logs, happens fairly quickly for most of the pathogens that we selected. But there are 2 populations here. So there's the initial inactivation, but then there's another population in all 3 of these studies on all 3 surfaces that seems to be hardier. So we're looking into what the potential residual risk can be from those remaining pathogens that are resistant. And the other thing that we also would want to consider in thinking about the indoor environment and possibly contaminated surfaces is transmission. So, wow, that was fast. Are humans more efficient transmitters of antimicrobial resistant genes than the environment? While a lot of the focus, again, has been on measuring resistant genes in the environment, we haven't really studied this indoor transmission dynamics very well. So QMRA, or quantitative microbial risk assessment, generally deals with environmental exposures. But there's another part of that, and that would be the secondary transmission. And it's quite possible that the secondary transmission is more dominant than the environment. This study that I've depicted here that I don't have time to go through is based on a colleague's work in Ecuador, Dr. Joe Eisenberg at University of Michigan, where he tracked the emergence of antimicrobial resistance in a population after a new road was constructed that ultimately introduced antibiotics to their chicken farming practices. So while the study focused on the emergence of antimicrobial resistance, it's a really rich data set that would allow us to identify the origin and whether this resistance is a result of colonial expansion or horizontal gene transfer that would help us get at this question about whether we need to focus more of our efforts on human-to-human transmission. So I'll summarize with the big research gaps that I see in this area for risk assessment. And one would be an emphasis on the spread and determining the sources, which sources or modes of transmission are most dominant, so that we know where to focus our efforts. There are reservoirs and amplifiers in the environment. We also need better dose response models for antimicrobial resistant pathogens, which are virulent factors, and we know that we often see orders of magnitude difference when we look at ID50s for virulent pathogens versus less virulent pathogens. We need to also look at horizontal gene transfer in dynamic systems. So this would include the human gut, but also in the built environment, as Amy alluded to, so that we can understand what's happening in water distribution and wastewater treatment plants. And then finally, quantifying the health outcomes across a community. Like the study in Ecuador, there aren't very many places where you can measure that type of data or take those types of measurements without the effect of other random effects. So more studies like that would allow us to identify where we really need to focus. Thank you.
I get the fun job of wrapping everything back together and talking about some other research in here, but as a modeler, I put 0 equations in this presentation, so everyone should be happy about that. in reality, what I'm trying to talk about is how do we build smart models to try and get to a point where we can start answering some of these bigger questions and get that information to a utility as quickly as possible, or have a tool that the utility can then use as they go about their monitoring of their system and understanding more about their system. So first of all, we want to talk a little bit about drinking water infrastructure. I'm not going to rehash everything that our great presenters have already talked about, but there are a couple things that I like to always put water in perspective about, and I put them in just really very generic terms that everyone's going to understand. The rule of threes of survival. You can last 3 minutes without air, 3 days without water, 3 weeks without food. I used to smoke. I can probably last about a minute without air. So obviously, it's going to be different from one person to the other, but it's a general rule of thumb that we can kind of say. And the other thing, kind of harkening back to when we used to do bioterrorism-related research, drinking water affects an entire population at one time. So you can have isolated incidents within distribution systems. It's really interesting when you get into the hydrodynamics of that. But in general, you can reach out to an entire population by being able to affect drinking water for good or for bad. So we really need to be able to kind of keep this into perspective. And when we wrap this up at the end of this presentation, we'll see all the other impacts that we can have beyond just drinking the water. So in general, we like to think of drinking water infrastructure in very nice ways. We drink this, it keeps us alive, so we don't want to think of it in the more nitty-gritty details and less nice parts that we'll see in the next slide. So this is a bit of advertisement for Philadelphia that's always used, Fairmont Waterworks, one of the first filtration plants in the country. And it's a very nice view of how we treat our water and what the water can look like, and a very nice picture of it. We see new infrastructure being placed in, some retrofits happening. And we can kind of show where we're going, the future of our infrastructure, and we have education and training the next generation of engineers and scientists. This is Charlie Weir, and donations to his college fund are always accepted, but we can talk about that later. But we want to try and make sure that everyone gets an understanding of what they're drinking water, where it comes from, and its tortuous path. This is its tortuous path, and I am obsessed, for one thing, with Legionella, so you really have to keep my time because I can talk about it forever. The other thing is wood pipes. So this one that you see here, this is actually a main line that's going in Florida. It runs for more than a mile. It's planks of wood that are held together by steel straps that are bolted down. That's how a wooden water main can work. You can also retrofit a wooden water main so you can connect new infrastructure into it. So you keep the old wooden water main, you add a ductile iron pipe cap onto it, and then you put the metal onto the metal, and they're going to be okay. This is actually a really interesting set. This is actually a wastewater pipe, but I just like this picture because this used to be a 3.5-foot trunk for sewer main. And this is primarily calcite deposits that have come up, but this is also a good illustration if you really want to illustrate artery hardening and things like that. So you went from 3.5 feet to about, I think the final size is about 7 inches in there. Anyway, I can't remember what the final inside side. Another wooden pipe, because I'm obsessed with wooden pipes, and then lead lines coming into the water meter in a household. So we have old, aging infrastructure, really, really old technology, and we have the results of forcing people to use lead pipes for us to be able to even connect them onto the distribution system in the first place. So if you want to talk about shooting yourself in the foot, we did that a lot over the years. So now we're going to get into less depressing stuff and just slightly more scary stuff on Legionella. Now, what I'm going to talk about in this case is primarily what Legionella is by way of a water threat, as well as what we can do by way of modeling it. So Legionella is not particularly emerging. We knew about it in the 70s. If you take it all the way back to Pontiac fever, in that first instance, we know about it from the 60s. But we're seeing it as a neglected health threat, is how I kind of term it. Now, we see a big pop-up of it around 2001, and it keeps increasing as we go into the present day. Now, this could be an aberration of sampling. Assaying for Legionella has become a little bit easier. If you're running it on a genomics unit like a qPCR, it's going to be a lot easier than it was back in the day. However, this is still a statistically significant increase in overall disease burden from Legionella in drinking water outbreaks. So just to kind of put it in context, Legionella is becoming even more of a concern as we go forward. And then when we talk about crypto next, we don't have as interesting of a plot, but that's also increasing too. So you're not really supposed to read everything that's on here, but the basic idea is this. We have limited information right now that we can pull from the open literature on the specific water quality characteristics that allow for Legionella growth and persistence. Like Amy went through already, so I'm not going to rehash that, about iron and organic carbon. Those are two data sources that we were able to draw from. Unfortunately, we couldn't use organic carbon in this model that I'm going to talk about because it wasn't conducive to modeling. But we could have a concentration of iron in the water, time and temperature, time being time in system in a distribution system. So the basic idea is this. If we have some way of modeling the growth and persistence of Legionella in a biofilm, we run that through a two-dimensional simulation because this is going to allow it to be much more specific to whatever utility is going to use this in the future. And then we're going to have a growth and persistence model on the back end that we can use in microbial risk assessments and other people can use however they're going to be using it. Now, big, massive, massive caveat. This doesn't have any mention of protozoa predation in this model. That is a massive problem. When you get blooming of Legionella in a biofilm or any environment, is because it has been taken up by a protozoa and essentially turned it into its own little tiny incubator. It's just one of the really cool things about Legionella. We do not have the data that we can model that process with yet. Give us about a year or two and my lab will get that out, but for now we don't have that information. This is just to basically build out what a structure of a model would end up looking like and to try and get some preliminary inferences off of it. So, basic idea behind a two-dimensional simulation is that you have variable variables and uncertain variables. Something that contributes to variability and something that contributes to uncertainty in whatever model you're working with. So we consider time and temperature to be variable, and we consider iron concentration to be uncertain. The other value of a two-dimensional simulation is that I can just keep this crunching until I get the value that is the actually correct value. So if you think back, all the way back to basic statistics, the law of large numbers, you run something long enough, you will eventually reach the correct solution. So that's the real value of running it through a simulation like this. This, you're seeing about 20 million calculations, and it takes about 20 seconds to run on just a bare bones MacBook. But the value of this is that you now have a distribution of predicted Legionella concentrations in a biofilm given whatever your characteristics are for iron, time, and temperature. So time in the system, temperature that your system's running at, and the iron concentration in your system. So then if you're at a different utility, you pipe in their values, and then you get their distribution of likely Legionella values. This model, as it is now, will never be used because it does not have protozoa predation in it. It's such a lacking hole that this would never be distributed out for people to actually use. This is kind of highlighting why one thing that looks like a small knowledge gap or a small data gap from initial glance actually kills the model in overall usability. But one of the values of setting up this overall framework is that we can now be adaptable. So all I did was I changed the temperature in the simulation, and now I have a very different overall profile of the growth and persistence. The other thing to know when you're reading one of these plots is the more shallow the curve, the higher the uncertainty. The wider apart the lines are, the higher the variability. So we don't have a lot of variability, and we don't have a lot of uncertainty. And you can kind of just keep playing around with this and watch as the curve shifts around. But you have to be a monumental computational dork like me to actually want to do that. So, but you do have that availability. So now what we're going to talk about, we're going to kind of move into another water pathogen. In this case, it's going to be Cryptosporidium. Because what we were talking about in the case of Legionella is a model framework that once we have enough information, will actually be a very usable tool by utilities to be able to say, I have a Legionella problem, this is what my Legionella problem looks like, and now I'm going to go through a risk analysis and build out all these different scenarios for how I can change that. And I'm going to show you how we can do that. So, if we talk about Cryptosporidium, it is a gastrointestinal pathogen, and you can be exposed in a number of different ways. In this case, this was a recreational outbreak. So it was a spray park in upstate New York, and a number of children came down with cryptosporidiosis. A spray park is essentially, it's a bunch of spray nozzles that spray water all over the kids who are recreating, and a lot of after this study, I'll never have my kids ever go to, go to one. But what you can see on the epi curve is they first visited the park, and then there was a lag, and then they had onset of symptoms. So there's an incubation period that any organism is going to have, so we have that in there. We can use this to back calculate out what they were likely exposed to by way of a concentration of oocysts, but the real value and the muscle of Chiomare gets leveraged when we start looking at design changes. So this was their original design. These are descriptive diagrams for a Markov chain model. That's what the lambdas are, loss rates in the Markov chain model. And this is their original configuration. So spray pad goes into the first tank, goes into a filter, they add chlorine, let the chlorine react, comes back to the first tank, and then goes out to the spray pad. So if you're an engineer, you're probably wondering, why do they have this pipe here, which we eventually called the bypass pipe, because nobody would actually want us to call the dumb pipe, because what you're doing is you're taking clean water, mixing it with dirty water, and sending the mixture of clean and dirty water back out to the spray pad. So all of our design change recommendations and all the scenarios we built removed this bypass pipe from the system, and you ended up with a very good reduction in risks overall. We ran through a lot of scenarios. We looked at filtration, then UV, then chlorination, then reaction time, then send it back out, a whole bunch of different configurations. I'm not going to go through all of this with you. If you want, it's this paper that goes through most of them that we went through. But basically, if you leave it all alone and don't do anything, you have a 60% chance of everyone getting sick if there's another fecal event on the spray pad. So it's kind of like a worst case scenario. Then if you just remove the bypass pipe, you have 100% risk, and that occurs about 50% of the time. So you have a 10% reduction in your highest risk if you just remove the bypass pipe. Once you start putting in all your other treatments, this highest risk ends up hovering around 20%. Still not something you want to see, but this is as though a child had an actual fecal accident on the spray pad, nobody caught it, it went down the drain and then went into the system. So this is worst case scenario, because that's actually what happened in the case that we were on. So now we need to kind of bring it all back into where we're at on where we kind of started from, because we were talking about the indoor environment, and then we dove really deep into water for a while there. So I had to go really quick on this, so it's a good thing we're recording it. So you can pause this and look at this big mess of a diagram. These are 3 independent Markov chains for fomites. A fomite is any inanimate object that can harbor a pathogen. So if I touch the desk, I've contaminated a wood fomite, doorknob, you contaminated an aluminum or steel fomite, and so on as you start contaminating everything. This model is you have one person who's contaminated the room, 2 subsequent users have come in, so just think of it as a shared office workspace or like a computer lab. And the value of it is that we can not only get an estimate of each of the fomites' risks presented to the subsequent users, but then we can combine the risks together into an overall actual risk that they're exposed to. So you're not touching just one fomite and then having a risk from that. You're touching all the different fomites in the room, and that combined together is what your actual risk is. What we're doing now is we're testing this with multiple types of gastrointestinal pathogens. Can you merge them into one combined risk? Can you do this with other different types of exposures as well to try and see where exactly can we go from here? Now, where this gets a very good link to the drinking water as well, that everyone probably knows every time you flush the toilet, you get this beautiful aerosol of toilet water that goes up into the air. If you close the lid, then you get this side stream that kind of hits the walls and then goes up and makes this beautiful aerosol. You're going to be contaminating the indoor environment with drinking water quite often. So every time you open a tap, every time you flush the toilet, every time you take a shower, you're still contaminating. And then you're going to contaminate the interior space, like Michael was talking about, with the skin cultures that were coming through as well. So you have to be able to think of the overall microbiome, not just from the different matrices that we typically like to house ourselves within, water, indoor air, indoor spaces, things like that, but it has to be a grouped exposure and a grouped overall risk that we're seeing. I'm going to leave this slide up here because this is what we want to try and do to try and spur on some conversation during the panel discussion. As you'll see, these are all need statements. So we need to do this, we need to address that, and so it's kind of a way of poking at the bee's nest and seeing what conversations we can get going and try and inspire some discussion for the panel ahead. Thanks.
All right, thank you. I'd like to invite the rest of the speakers up here. And then we'd love to hear from you, comments or questions that you have based on what you've heard today. And Sarah, if you give me a 2-minute warning as well. Okay, yeah. Yeah.
I've got one question. So I'm a sustainability coordinator. I work at Beloit College, which is in southern Wisconsin. We're looking at the well building standard. I'm not sure if any of you are familiar with it. But if you are, I just kind of want your feedback on, building standards such as LEED and this newer one that is focused on the occupant's health within a building. What are your recommendations for, working with the building standard, and are there opportunities to go beyond those building standards to make sure that we have adequate control and sanitize surfaces within a building?
So from my perspective, so I'm focused a lot on mold exposure in housing, and I feel like a mold standard is actually something that we're still working towards. This doesn't necessarily it's not directly related to the well building standard, but I think we need to start developing these building standards so that we can have these places as sort of checkpoints, so we can have something we can move towards. And all of this goes into that design question of, I really like Amy's point about, if you don't design it, we're going to unintentionally expose ourselves. And I think that applies to water as well as buildings. So it's definitely something that we need to focus on. And for one question, any other comments on that?
I was just going to add that, with the conservation features in buildings, a lot of times it can be a simple fix. So some automated flushing in the system seems like it's wasting water, but really it's just a small amount and it's worth it for the public health. So with all these standards you're talking about, I think making sure that public health is part of the equation. So flushing, automated faucets, sometimes there's something about them that can attract pathogens, maybe the mixing valves, the temperatures. If we understand what's creating these problems, hopefully there's some simple fixes with the design.
So one thing we didn't go into is we went from the microbiology and the engineering and dove straight into the risk modeling is a paradigm called hazard assessment of critical control points, or HACCP. You can either view it as kind of like a preliminary before a QMRA, or a QMRA can inform it. But either way, what you're looking at is what about your plumbing. So basically to what Amy was talking about, but it's in a computational kind of sense. What about your plumbing can you control? How can you control it? You basically just do an overall assessment of everything in the building. And one thing that we have seen in performing HACCPs in different buildings, some of which are hospitals or other facilities, is that it becomes, each building is different, and each building seems to be making its own ecosystem. So kind of like what you were presenting with as well, Michael. The building a standard is something that we can look towards and achieve, but I think one thing that has to be kept in mind as well is that each building is going to be different, just because of the occupants and the environment and the water coming in and everything else.
When you're building a new, I come from a background before I got into microbes of real estate and running large apartment buildings, and then I also had a consulting practice that worked with hospitals. So like the two things that I see when you're talking about this from that perspective is, oh my God, if you start doing the microbes in my buildings, I'm gonna get sued. So I think from a policy risk, you've got to consider how are you gonna address that. Then, from the asthma and mold and all of that in the apartment industry, if somebody said anything about mold, people were running for the door going, no way, there's no mold here. So, you have to figure out how can you work together, particularly in these older buildings. And, a lot of those are the lower income population who doesn't have the voice to kind of raise the issue. The other thing I would say is old college dormitories are full of mold, and we're not doing anything about that. So, when you think about how to set policy and address all this old infrastructure, you've got to figure out how to get funding to help, address the issues in this older infrastructure, because, they're not going to just do it out of the goodness of their heart.
So it is a challenge, especially when you're getting into hospitals, to get that buy-in and get them to agree that you're going to come through and look at everything and basically tell them where all the dirty sinks are and where all the other problems are. And from my experience, the large building operators know the difference between gross negligence, and other definitions as you go down the lower drastic endpoints, right? And so if they know, if you are a hospital, you already know that this is a potential problem, and therefore one of the ways of being able to address it is to find out where the threats actually are, which is where the HACCP comes in, and then eventually the other pieces to start solving this issue. So I think there are quite a few large building operators that are understanding that they have to get ahead of the curve on this, and so we're seeing that to an extent. Others may need convincing, but those trailblazers will start kind of showing that this is a value added to them, if not for anything other than it goes from gross negligence to negligence.
All right.
Yeah.
And for some of that stuff, I think you do need these kinds of standards as well, which would be really helpful, where you can get a clear yes or no. Does this building have a mold problem? Yes or no. Whereas now we don't necessarily have that. So it's a little bit trickier. It's a little bit more qualitative. I mean, there's some buildings where it's going to be very obvious, but a lot more in that kind of gray area. So I think moving towards a standard would definitely help with some of that, but then you also do need the buy-in of these people to help correct these problems that are going to cause health effects.
And also to that point, there's another interesting, so there's the healthy buildings standard that we're talking about, but then there's also the adaptations to the Safe Drinking Water Act that needs to be considered as well. So however the EPA decides they finalize on a consecutive systems language can end up affecting whether or not large building operators are going to be seeking this kind of information in earnest. So if you are adding anything into the water to try and mitigate your biofilm development or any of the pathogens in your water, depending on how EPA settles on the final language, you could be a consecutive system regulatable under the Safe Drinking Water Act. If that happens, then they got to figure it out ASAP before the rule actually gets enacted and they start getting regulated.
Probably not in the next 4 years.
Well, no. Well, I mean, it takes 5 to 6 years for the promulgation and everything to go into effect anyway. But so I mean, there's another aspect to it. So there's the initiative to try and ensure that you have safe and healthy buildings, and then there is the what are you doing to your water once it gets into your building kind of question, and they're both kind of working at the same type of concept.
I think your point.
Oh, sorry.
I think your point is an excellent one about liability, and at Pittsburgh, in Pittsburgh, which of course has been the site of a very well-publicized Legionnaires' problem at a different hospital than where we were. We were very lucky that the hospital was completely cooperative with our study, but I have been privy to another hospital and another study altogether where there was quite a bit of obstruction for exactly what you mentioned, which is that people, at least some stakeholders, would have preferred not to know what
Right, because
organisms
it's
were
discoverable
present.
information. If my child dies and you put my child in a room that had,
Yeah, I think from
I'm going after, somebody will go after you.
right, and so I
Which
think
is problematic because you can't solve the problem if you don't look.
scientists tend to think about a lot of these issues in a vacuum, and similar to whether it's the Human Genome Project or the Microbiome Project, there are interesting ethical and legal considerations of all this work, and that's a nice thing about a meeting such as this. It actually kind of brings things together. But I agree with your point.
Well, the other side of the coin here, we're focusing on the building owners. What about the water utilities, right? They regulate up to the property line, but the exposure happens in the building. And so the lead and copper rule is actually the only example where they monitor at the tap, and it's a shared responsibility model between the homeowner and the utility. And I think that is an example where we need more of that and something analogous for the Legionella.
But we just like, I live in Colorado, and there's some regulations about not testing your own water in Colorado.
Oh, that's very interesting.
Really? We talk
Is it
more
the
about
water
that.
law, Colorado water law?
Yeah, Colorado. We live in our own land when it comes to water.
Okay, so thank you for the talk. So now we know the microbiome structure where we are exposed to is very important. And also from my recent studies, actually the more they clean in terms of building types, actually the building that does a lot of cleaning, actually the microbiome structure is not that healthy. So especially, so what are your suggestions for solution, especially for those buildings that may have more vulnerable population, for example, hospital, like NCIU, that kind of topic. Other than like a really chemistry-based cleaning, what are your solutions or suggestions for having healthy microbiome in indoor built environment?
Do that first.
I think that's a really excellent question. I think that still is something that we have to answer. So I think we're moving towards this idea of what is a healthy microbiome, and it's probably not a complete absence of all microbes. So there's probably going to be some base microbial community that's either benign or even potentially beneficial that we could hopefully eventually try to design towards that, and hopefully remove the potentially harmful microbes. So I think it's still an active question. We still need more of these cleaning type studies where we look at what are these cleaning products actually doing? Are they removing what we think they're removing and what we want them to remove? And are they leaving behind what we want to see there? So from my opinion, I think it's still an open question.
And I think actually first we need to define what clean is. That's a risk question. So you want to determine how clean is clean, but also how clean is clean enough, right? So we need to be able to determine, like, is there really harm at certain levels? And then you can answer, are the products or are the technologies like meeting your intended level?
Yeah.
I think what you've just described fits extremely well with the concept of the hygiene hypothesis. Are people familiar with the kind of idea, which just, I think, sort of intuitively makes sense, but there's really good data to back up the fact that as civilization becomes more and more sort of advanced and cleaner, many diseases, such as infectious diseases, go away, but many other disorders are more common. And I don't know if Amy, you or Mark are familiar with really interesting work by the people that coined the term hygiene hypothesis. You probably are, but as I understand it, in Britain, one of the most interesting studies that I was aware of, because appendicitis is something I deal with and I'm really interested in, was that they sort of mapped out where in the United Kingdom water filtration plants, or whatever the term is, as they were more common in the countryside of England, appendicitis started becoming more and more common, and the maps overlaid really nicely. So I think that is exactly the idea of what you're saying. Now, why that would be the case in a building where you're actually cleaning it and the opposite effect arises, that's really fascinating, but it sort of makes sense that you're somehow selecting for particularly fit or hardy organisms.
And what I'd add to that as well is that, we definitely need to know more about cleaning. We need to know how clean is clean, or how clean is clean enough, because you can't clean constantly all the time. But the other thing is, how do we verify the level of clean? So right now, the typical protocol in a hospital is you'll do ATP swabs. So you'll take a swab, you put it into an ATP unit, you get a value that correlates somewhat to a indicator organism. What does that mean about pathogens? Is ATP even that good of a test anymore? Are there better options? So there are some questions on the validity of verifying the cleanliness at the same time. And whether or not what you're doing by way of looking at those indicators is even indicative of pathogens at this point.
All right, well, I think we're out of time. If you have more questions, we'd love to talk to you afterwards, so please come up and say hello. But thank you, everybody, for coming. Please make sure you signed in, and we'd love to get your feedback on the evaluation form as well. So thank you to all of our speakers, and thank you.
Thanks.